The overall goal of this procedure is to quantitate neutrophil extracellular traps, or nets, released from human neutrophils on a high-throughput scale.
The methods presented here can help answer key questions in the fields of inflammation and autoinflammatory diseases through the study of the mechanism and regulation of in vitro neutrophil extracellular trap formation in human neutrophils.
The implications of this technique involves identification of essential net-mediating signaling pathways and net inhibitory or net-stimulating molecules.
To isolate neutrophils from blood, aliquot 20 milliliters of human blood into 250 milliliter conical tubes. Add 10 milliliters of 6% dextrin, then gently mix and allow the solutions to sit at room temperature for 20 minutes. Carefully transfer the leukocyte-rich upper phase into clean 50 milliliter conical tubes and use sterile PBS to fill the tube to the top. Then centrifuge the sample at 400 times G for 10 minutes and use 50 milliliters of sterile PBS to resuspend the pellet. Repeat this wash step one more time. Next, prepare 5-step Percol gradients of 85, 80, 75, 70, and 65% in two 15 milliliter conical tubes, and layer 2 milliliters of the leukocyte suspension on top of each gradient. Then, centrifuge the gradients at 800 times G for 30 minutes with the brake off. After centrifugation, collect the cells in the layers at the 75